with that, the panelists, let's start with the panelists. We are open to questions. And if you should be able to unmute yourself, if not, the office will unmute yourself for your questions and discussions and comments that you may have.
I have a question, if I may start. This is Matthias, and feel free to switch on my video also. I somehow can't do that. Peter, that was beautiful. It was a great overview. I really enjoyed it, and I always, still learn something when you give a talk like this. It's amazing. I have a specific question about macrophages in the plaque and also in arteries. You mentioned that now with single cell RNA-seq there is a multitude of subsets and new cell phenotypes to look at. I wonder what your feeling is. What is a reasonable resolution? I'm struggling with this. So we used to think about macrophages as one cell class. They're not lymphocytes, they're not neutrophils. Then we started to think about recruited versus resident. But now I think you can play that game until you have 20 macrophage subsets, and there is probably a reasonable number that's higher than 2. I wonder what you think about this.
So I think with single cell, we're in a very descriptive stage. And I wrote a review with Joran where I put in a paragraph where I said that sort of like Lord Rutherford said, in science, there's physics and all the rest is stamp collecting. I think we're in the stamp collecting phase of single cell. And I evoked the pitched battles among the academics in the Middle Ages, who in the scholasticism school spent years debating how many angels can dance on the head of a pin. And I think that that's where we are. Now, Joran read this draft, and he told me that that was too controversial and flagrant, and that I should remove it, which I did. But then when the paper came back for revision, I put it back in. And he didn't notice. So it's actually in print. So that's what I think. I think we're in a descriptive phase. Everyone is playing with the fun tools. It's the fad these days. It's sort of like if you have a seahorse device, everything is metabolic. And there's a 10x machine. So now everyone can do their beautiful UMAP plots and describe all of the populations they get. And you're right, it's a game of infinitesimal calculus. The challenge is really to get serious about function and to test in critical and rigorous ways what really makes a difference. So this debate about the angels dancing on the head of the pin is where we are, and that's what your question really throws into stark relief. And so that's a challenge for the field. We need to move past simple categorization and get to function. I think some of the people on this panel actually are working in that direction.
Great, thank you. I think Dr. Klaus, do you have a question, sir, a comment?
Yeah, this is Klaus Ley. Hey, Peter, a great lecture. Can I get you to look into your crystal ball? And we know that colchicine works. We know that canakinumab works. Probably IL-6 antibodies will work. Is there room for combinations? Where do you see this field of anti-inflammatory therapy going?
Well, I think if we're going to combine, we should look at targeting orthogonal pathways, right? So perhaps colchicine is working at the level of the granulocyte, and some of the proteomic data from LODOCO2 suggests that although there's this rumor that colchicine inhibits the inflammasome, the data are really not that rigorous. And the proteomic data from LODOCO2 suggests that it's working on granulocytes. And as I said, in granulocytes, we think that they may be participating in some forms of acute coronary syndrome. So I guess we need to, if we're going to combine anti-inflammatory and immune interventions, we should do it orthogonally rather than pile on the same. The idea of targeting IL-6 is that by going one step distal to interleukin-1 beta, we can leave interleukin-1 beta's role in host defenses intact, but attack really what we think is causal in human atherothrombosis. The other way around this is, of course, to target more precisely. And when I give a full lecture about clonal hematopoiesis, I show the landscape of human leukemia and the genetic defects which have been identified through the decades. And I have a big arrow about 22 years ago when imatinib, Gleevec, was approved on the basis of a genetic target. And where are we in the vascular field today with genetically targeted therapies? Zero, okay? Maybe in amyloidosis in cardiovascular medicine, we have some genetically informed therapies. But in the vascular atherosclerosis field, we have nil. So we're more than 2 decades behind oncology. And perhaps things like CHIP will allow us to identify people who respond to particular interventions. Perhaps in JAK2, V617F, which has a very elevated cardiovascular risk, perhaps we can use a JAK inhibitor. In TET2, we may be able to use an inflammasome IL-1 beta or IL-6 inhibitor, and down the line. So I think that number one, I would use orthogonal pathways if I'm going to combine agents. And number two, I'd like to really stop talking about the buzzword of precision medicine and get there. And certainly many people are working towards that end, including our group.
In that spirit of better targeting and more specific, you didn't mention the adaptive immune response. Do you think the autoimmune response to ApoB and other atherosclerosis antigens plays a role?
Well, you're going to tell us. I mean, your group is very far along, and you know, it's interesting. You started with the tetramers, and now the tetramers are out of fashion. You've superseded that. But your group is probably one of the farthest along in the clinical translation, or we're preparing for the clinical translation of targeting adaptive immunity. And of course, there are other groups as well at UCLA and in Malmö. And so we haven't got the clinical fruit yet, but I certainly do believe in the operation of adaptive immunity. As I said very early on, I think the T cell mediators, and I've written about this, is I think the T cells are the generals and the mononuclear phagocytes are the infantry, that the mononuclear phagocytes are the orchestra members, but that the T cells are the conductors, or the T cell committee get together to orchestrate the innate immune response. So I am definitely a believer in causality of adaptive immunity, and some of the early work that we did in transplantation arteriosclerosis, I think bore that out. Where we were in one of the very first experiments with knockouts, we were able to show that gamma interferon is causal in experimental allograft arteriopathy, but not in the mesenchymal, in the parenchymal rejection in allografting. So I definitely have believed for decades in the importance of the adaptive immune response, and wish you well, Godspeed in your effort. Please do it in time for me, all right?
Peter, question about CHIP.
Oh, Chris Valentine, yeah.
So, you know, we tend to lump this together, but as you just mentioned, DMT3A is the most common observed variant in terms of the driver gene, but it really has a very, in fact, the more you look at the data, the weaker it appears the association with events. So TET2, ASXL1, JAK2. So we're probably going to have to, and then when they looked at the epigenetic changes, they were in the opposite direction. DMT3A was very, very different from TET2 with this. So I guess this is the next step when you talk about precision. We're going to have to look at each of these, take this huge data sets, but looking at probably a lot more detail at the specific driver mutations to understand fully what's going on.
Yeah. So what you're saying is, if you see one chip, you see one chip. And Christy, we have a paper in press together that is suggesting that there's a particular diathesis towards increased peripheral artery disease in the DNA damage and repair, these such as PPM1D and TP53. And so that's wonderful work that we enlisted you in, and
Right.
that I think
I thought Derek's paper is very impressive that it's not uniform, and we'll have to look at these probably a little bit more carefully, don't you think?
Yeah, so that paper is now in press, but it's available in preprint form. Derek Claren led that, and you participated in it, and I think it's very, very informative and very interesting that there's a predilection towards PAD with the DNA damage and repair mutations. And I agree with you about DMT3A. The AXL1, it turns out that there's a PCR artifact in the genotyping that's been done in UK Biobank. And I think the jury is still out on AXL1. So there's a very recent paper on the fine points of chip calling by Alex Bayek and Pradeep and Sid Jaiswal that I think some of the differences in the publications about the degree of increase in cardiovascular risk with chip mutations can be accounted for because of technical issues and the population.
Right.
Everyone mines UK Biobank, and remember that UK Biobank is predominantly Northern European, almost all Caucasians, and well enough off to volunteer to participate in the building the biobank. And certainly the patients that I see and patients that you see also in your environment don't fit that profile.
Correct.
We see people from more disadvantaged backgrounds and with much greater ethnicity diversity than in UK Biobank. So we all use the UK Biobank, but we should not pray at its altar.
Wonderful, thank you. So we have a couple of questions online. I'll just take one of them. Another distinguished scientist, physician scientist, Dr. Tagmeyer. His question is, fabulous journey. Quick question. What is the effect of diet, intermittent fasting on interleukin-6 and bone marrow transformation?
Yeah, so thank you, Heinrich, who's been a colleague and a wonderful friend for longer than either of us care to admit. And he has the small lab with big ideas and was the last graduate student of Sir Hans Krebs and has wonderful anecdotes about that, which he's written about recently. So thank you, Heinrich. I think that lifestyle is primordial in cardiovascular prevention. It's a battle that we're losing globally. And I think that certainly there's evidence that the Mediterranean diet can lower biomarkers of inflammation. People have not focused specifically on IL-6, but I think that that's a very interesting question. But, I presented a talk at the Italian Society for Cardiology, and the question that came up from the audience was, well, now that we whipped LDL cholesterol, what's causing all of this? And I said, it's the global pandemic of obesity, which is going to be driving cardiovascular risk. And unfortunately, we're sowing the seeds for a whole disaster because of the obesity in childhood with concomitant dysmetabolism. So I'm very concerned, and I think that diet and physical activity are the foundation of cardiovascular prevention, particularly in youth. Although perhaps the application of polygenic risk scores will give us a handle, again, in precision medicine, in who should get more intense intervention in youth. But we're never going to have clinical trials looking at outcomes that in therapies that are allocated on the basis of polygenic risk scores in youth, because no one will fund them and no one will live long enough to see the results. So thank you for that question, Heinrich.
So final question by Dr. Zhilong Jia. Wonderful report. Are there other anti-inflammatory targets for the atherosclerosis except for interleukin-1 and interleukin-6? Thanks.
Yeah. Well, as I said, the inflammasome is being targeted, and there are dozens of cytokines. We did experiments with neutralizing CD40 ligand back in the dark ages. And there are many pro-inflammatory cytokines which can be targeted. The trick, though, is to find those that are causal and those whose neutralization or inhibition will not impair host defenses will not impair tumor surveillance. You know, when we went to Basel, Dr. Ricker and I, back in 2009, to try to convince the leadership of Novartis, we call them the gnomes of Basel, to undertake this experiment with us. And to their eternal credit, they did. But the questions that came up were, number one, are we going to be reactivating tuberculosis or opportunistic infections? And that turned out not to be the case in CANTOS, targeting IL-1 beta. And they were concerned about whether we would be precipitating ventricular rupture or the other mechanical complications of acute coronary syndromes. And in fact, we didn't. And in the era of primary PCI, these complications have become much rarer. And experimental work that we did with Phil and Matthias, neutralizing IL-1 beta, we saw actually improvements in myocardial remodeling, not adverse effects. And so I think that we have to be very careful. You have to get into thousands and thousands of patients studied for many years in order to see these infection signals. And so what we need to do is to really target the causal mediators and be very wary about impairing host defenses. And then tumor surveillance was the other thing that came up in our discussions, or I brought it up in our discussions with Novartis back in February 2009. And of course, as people know, actually we saw a reduction in cancer. And in fact, that is why the study was allowed to continue to completion despite the excess infections, because there was at the highest dose tested a 77% decrease in lung cancer deaths during the course of the trial. So our concerns about impaired tumor surveillance were not germane. And actually, one of the reasons we don't have canakinumab as a drug for cardiovascular medicine is because the sponsor ran off to do a bunch of cancer trials. They were so enamored with the exploratory analysis that we did on cancer. And there's very good biology. I've written about with Sebastian Kobold, who's an oncology colleague from Munich, from LMU. We wrote a piece about the commonality of the tumor microenvironment and the atherosclerotic plaque, and how, although very upstream, we think that cancer is more somatic mutations than atherosclerosis, but we're learning more about the somatic mutations that drive athero. But once you get the disease started, the pathways and the mediators are very similar. So I was not surprised, or I was ready to explain some of the benefits for cancer metastasis on the basis of work that we did on inhibition of metalloproteinase expression by limiting IL-1 beta signaling back in the 1990s. So I think there are many targets, but we have to choose them judiciously.
Thank you very much. With this wonderful webinar, Dr. Eugene Braunwald, MD Lectureship, comes to its end. I'm grateful to our panelists, Dr. Yibing Wang, Dr. Christy Ballantyne, Dr. Klaus Levy, and Dr. Matthias Narendorf. And Dr. Libby, our sincere gratitude to you, our admiration to you for what you have accomplished in this beautiful journey that you described to do. Thank you all very much.